Today, our presentation will be on the hammerhead ribozyme by
Poe
Alan.
and Hossein.
Our understanding of biological catalysis has been extremely limited, especially up until the last 25 years. In this time span, many catalytic RNAs were discovered and characterized. Of the ribozymes discovered, the hammerhead RNA was one ribozyme that was found in the satellite RNA of tobacco ring spot virus. This hammerhead ribozyme will be the focus of this presentation. So, the discovery that RNA can sometimes be an enzyme suggests that by understanding ribozyme chemistry better, we might be able to learn more about how life may have originated from a world of self-replicating ribozymes. So, by understanding the chemical processes of the hammerhead ribozyme during the RNA catalysis reaction, the significance of the molecular structure and atomic interactions in biology can be observed. The things this presentation will go over include what a ribozyme is. Then we will talk about the specific ribozyme called the hammerhead ribozyme and go over its structure and its chemical usage in cleavage. This will give us a better understanding of the ionic properties of the hammerhead RNA and how the ions affect the RNA backbone. Eventually, this will lead into how we can use the hammerhead ribozyme and our current knowledge of it in real-world applications, especially in medicine. So, what exactly is a ribozyme? First off, let's give some background to the ribozyme. At first, it was believed that any and all enzymes were just proteins. This was widely regarded to be true until 1989. That year, during the observation of a splicing mechanism of an rRNA intron from protozoa called Tetrahymena thermophila, researchers Thomas Sech and Sidney Altman discovered that no proteins were actually involved in the process they were studying, and that some RNAs could be used for the catalytic functions. Usually, the catalysis happens before the transcription phase, but this post-transcriptional process of rRNA led to the discovery of ribozymes. Eventually, it became understood that ribozymes were special RNA molecules that could adopt complex tertiary structures and serve as biological catalysts for RNA. The hammerhead ribozyme, specifically, is an RNA module that catalyzes reversible cleavage with a specific structure. Overall, the hammerhead ribozyme consisted a minimum of at least 3 base paired stems flanking a central core of 15 conserved nucleotides. But the most important parts of the hammerhead ribozyme for the chemical reaction include its active site, the metallic ions, and the nucleobases used.
What makes the hammerhead ribozyme special is that it is extremely small and crystalline, and the hammerhead ribozyme can accept monovalent and divalent metal ions. Although the monovalent ions can only be used in extremely high concentrations, it shows a flexibility that other ribozymes do not have. Also, it uses a guanine 12 base and a guanine 8 base to perform the catalysis. Therefore, although all ribozymes catalyze reactions, the hammerhead ribozyme's small size, thoroughly investigated cleavage chemistry, known crystal structure, and its biological relevance in humans makes the hammerhead ribozyme particularly well suited for biochemical and biophysical investigations into the fundamental nature of RNA catalysis. The hammerhead ribosome uses two magnesium 2 plus ions in order to perform four main jobs. The structure of the ribozyme allows for these two magnesium ions to bind in at the perfect position to enhance guanine 12 as a base, enhance guanine 8 as an acid, stabilize the negative charge of the oxygens surrounding the phosphorus, and align O2 and O5 as the axial bonds of the trigonal bipyramidal structure of the central phosphorus. The structure of the hammerhead ribozyme allows for these four actions and the subsequent efficiency of the reaction because of them. Due to the first magnesium ion bonded to the guanine 12 base, the shared negative charge between the nitrogen 1 atom and oxygen atom in the guanine 12 base is focused on nitrogen 1. Thus, the guanine can act as a base at the nitrogen 1 position to accept the H plus ion from the oxygen 2 position of cytosine 17. This yields an oxygen 2 that has a negative charge. The now negative oxygen 2 moves to bind to the phosphorus underneath in order to relieve its unfavorable charge. The magnesium 2 plus ion in the second bonding site causes the shared negative charges of the oxygens to stabilize to two negative charges. This leaves the bond of the phosphorus with the oxygen 5 below to become the weakest out of the 5 oxygen bonds. Meanwhile, the magnesium ion also weakens the bond of oxygen 2 and hydrogen 2 in the guanine 8 base, essentially enhancing the nucleobase as an acting acid that donates protons. The hydrogen 2 can then be accepted by the oxygen 5, and thus the bond is broken and cleavage occurs. The second magnesium ion also situates the oxygen 2 and oxygen 5 as the axial bonds within the trigonal bipyramidal structure of the phosphorus. These are the two weakest bonds due to the fact that the axial bonds have three 90-degree bond angles each, whereas the equatorial bonds have only one each. By placing the two bonds of oxygen 2 and oxygen 5 in the weakest positions, the catalysis is performed even more efficiently by the hammerhead ribozyme.
Currently, our knowledge of the hammerhead ribozyme shows us that every ribozyme has the potential to have a workable state that can be targeted towards medicine. They may eventually play an important role in the therapeutic agents, biosensors, and its application in functional genomics and gene discovery. Also, they have been used against oncogenes and viruses and have helped in the understanding of the role of genes in developmental processes. Some researchers have even begun using hammerhead ribozymes in clinical situations targeting viral diseases such as different strains of HIV, hepatitis B, and hepatitis C. Although our understanding of ribozymes still has their limitations, ribozymes are almost 1,000-fold less efficient in catalyzing comparable reactions in comparison to protein enzymes. And even though ribozymes can increase the occurrence of reactions by 100 million folds, ribozymes cannot compare with proteins as multiple turnover enzymes, mostly because protein product release is so slow that the catalytic site of the ribozyme is easily saturated. Also, when trying to apply hammerhead ribozyme intracellularly for the therapeutic purposes, the requirement of elevated concentrations of metallic ions to obtain high catalytic activity becomes an issue. For example, the intracellular magnesium 2 plus ion concentration is known to be approximately 0.8 mmol per liter. much lower than is commonly used for in vitro reactions. Some other obstacles include the efficient delivery to a high percentage of the cell population, inefficient expression of the ribozyme from a vector or intracellular ribozyme concentration, co-localization of the ribozyme with the target, and specificity of ribozyme for the desired mRNA. Also, crystallography cannot tell the difference between metals, specifically magnesium and H2O atoms, because they both have the same number of electrons, the magnesium plus 2 ion containing 10 electrons, and the water molecule also containing 10 electrons. That is why manganese is sometimes used instead. In summary, hammerhead ribozymes have a specific structural and elemental difference that other ribozymes don't have. Specifically, the magnesium ions contained in its structure is used in two binding sites to enhance the base, enhance the acid, stabilize oxygen charges, and axially position oxygens. The guanine 12 base in the hammerhead ribozyme acts as a base, while the guanine 8 base acts as an acid in the catalytic reaction. However, we still don't know exactly which bonds are needed to be used in the catalysis. Although we know that RNA itself must be an active participant in the chemistry of catalysis rather than serving merely as a metal ion bonding scaffold, we have not reached the level of understanding of ribozymes to make it applicable to real-world problems. As more research is conducted, science might give a better understanding of the gene regulation of DNA. Eventually, when we develop a better understanding of hammerhead ribozyme processes, we might be able to manipulate and alter the chemical makeup of ribozymes for future uses. Thank you for your attention, and this concludes our presentation. Do you have any questions?